This presentation is about CRISPR and Alzheimer's-related proteins. My name is Madison Strange, and I was responsible for information on CRISPR background and how CRISPR works. And the other members of my team were Kathryn, Kathy, Sanvi, and Teo. So a little bit about CRISPR is it stands for Clustered Regularly Interspaced Short Palindromic Repeats. And it was actually derived from a genome editing system in bacteria, specifically E. coli. Francisco Mojica introduced it whenever he was sequencing one particular strain of E. coli and discovered that there were sequences between the repeats known as the spacer regions of CRISPR, and that matched the sequence of a particular virus. And scientists use CRISPR now to edit genomes. This can be used to correct genetic defects, treat and prevent the spread of diseases, and improve the growth of crops. And they do this by deleting or adding genetic material or replacing segments with a newly synthesized DNA sequence. How CRISPR works. So a virus is injected into the genome of a cell, and the CRISPR sequences are transcribed into RNA sequences, and they act as a sort of guiding sequence to the matching sequences of DNA. So the RNA segments target the virus's DNA, the CRISPR gRNA binds with the matching viral DNA, and then the enzyme that is a part of the CRISPR system is called Cas9, and it cuts the DNA at that particular sequence. And this cutting of the enzyme allows bacteria to remember the virus is injected into the genome and disabling the virus whenever it's injected in the future. What is Alzheimer's? Alzheimer's is a progressive neurological disorder that causes brain cells to die and which eventually leads to parts of the brain shrinking. Neurons lose connections and eventually stop functioning. Alzheimer's affects a person's overall mental state by decreasing their thinking and behavioral skills. So what causes Alzheimer's? Alzheimer's is caused by the abnormal buildup of amyloid and tau protein deposits. Amyloid protein deposits form plaques around the cells, while tau protein deposits form tangles within a cell. These deposits cause loss of neuron functions like neuron communication and metabolism and repair. Genetically, there is not one specific gene that causes late onset Alzheimer's, but there is a variation of a gene called APOE that can increase a person's risk for developing it. For early onset Alzheimer's, there are 3 mutations that are associated with it, one of them being a mutation in the amyloid protein gene.
Well, let's talk about where did the research happen. Experts at the University of Tokyo in Japan identified a new protein pathway that leads to Alzheimer's. And what the researchers did is that they used the molecular scissors of CRISPR to search for new genes related to Alzheimer's. And among the scientists who worked in this study, in the research, there was Yukiko Hori. He's a co-first author on the research paper published in Facet Web Journal, and he's also a lecturer at the University of Tokyo. Professor Taisuki Tomita, she's an expert in pathological biochemistry at the University of Tokyo, and she was the leader of the research lab that performed the study.
Now I'm going to explain how scientists use the CRISPR-Cas9 system that Madison described in their Alzheimer's research. So scientists grew mouse cells in a laboratory, and they used the CRISPR-Cas9 system to remove over 19,000 genes from these cells. And after they removed each of these genes, they measured the amount of amyloid beta that was produced by each of the cells, minus the gene that had just been removed. What scientists found is that one gene in particular, called the CIB1 gene, caused a significant increase in the amount of amyloid beta produced by the cells, and that is a protein that is typically found in cells. They found that this occurred because CIB1 typically attaches to gamma secretase, which is a molecule that plays a role in the processing of amyloid beta. Typically, CIB1 attaches to gamma secretase, and then gamma secretase is able to move inside the cell and in throughout the membrane of the cell. But in the absence of CIB1, gamma secretase was only able to stay within the inside of the cell, and therefore the processing of amyloid beta was affected. And as Sanvi mentioned, a buildup of amyloid beta protein is one of the indicators of Alzheimer's disease.
Now that we've used CRISPR to reveal one of the potential key players in AD, let's see how CRISPR can help us treat and potentially cure the disease. And for that, we'll be turning our attention to a popular protein E, or APOE. So for some background, the APOE gene is one that codes for a protein that transports lipids and other particular materials from the brain into the bloodstream. It is coded in 3 variants. We see that people that have E2 variant seem to show some sort of protection to AD, while those that have the E4 seem more likely to develop late onset Alzheimer's disease sooner. Now with CRISPR and the help of the Cas9 technology, we can cut out the APOE E4 gene and then replace it with the E2 variant with the help of a donor DNA. Now with that, of course, there comes the issue of ethics, and we've actually seen experiments that have been done prior, so we're not the first to look at the APOE E2 variant. Some people scientists have particularly used the DNA in a vector using an AAV to insert it into the brain cells and have seen promising results, though it does have its own setbacks.
But with the experiment using CRISPR and the Cas9 technology, you have to take into consideration that astrocytes, which are the cells that produce express the APOE and produce the protein, are cells that only regenerate when there's damage to the CNS. So we would have to most likely genetically modify undifferentiated cells in the embryo, which brings in another level of ethics and questioning that is still an uncharted territory full of minefields. But until then, we'll see what we can do and see how we can continue to further our research with the help of CRISPR.